Good morning. So, firstly, where is Michel? Where is Michel? Thank you very much, Michel van der Bank, for bringing me here. I'm very happy to be in the Kruger, very happy to be in Africa. This is a very nice picture of a system back home in Algeria. This is at the southern tip of Algeria, so this is Libya here, Niger. Mali is a bit off-site here. The last botanical survey here was done in the 50s. So we have no idea what plants are around there. And we definitely need to go back there. The problem is some crazy people now are going around here. The kind of people who want us to go back in the 7th century, you know? Anyway, so when they'll disappear, I'll let you know, and then we can go there and sample plants. Pulling this talk together, I had to think a bit to actually use the term barcoding in my talk. So we'll basically start little by little. Firstly, phylogenies. We have seen phylogenies shown by William, Crystal, and phylogenies are actually methodological frameworks that allow us to study evolution as a whole. And this is not new. So I used this during my PhD, and I'm still using this slide. These guys were working on allozymes in the 60s. And they actually made this very important point, is that a phylogeny by itself means absolutely nothing. It's just relationships. But now, if we can link the phylogeny with neontological data, organismal data, then we have proof of evolution. And since 65, we're talking about molecular data, right? Evolutionary processes, I'll try to briefly talk about it. We have biogeographical diversification, island biogeography. Everybody's familiar with the MacArthur and Wilson, for example, model. Adaptive radiations. Americans working on these Caribbean anoles. That's a case study. You know, we teach that at school. The African cichlids, there's going to be a great talk on the African cichlids during this conference. Diversity-dependent diversification, so basically diversification slowing down, even stopping because there's no more room. All the ecological niches are being filled. The tempo and mode of abiotic factors, ecological fixed filtering, etc., etc. And then obviously these key innovations. So lineages would evolve particular traits, and this is what Crystal was talking about, and these particular traits would allow these lineages to actually adapt, get fitter, and eventually diversify, but that's not always the case. Now, all this can be identified using phylogenetic trees. We can do that. We need, as the brilliant talk by Crystal, ancestral ecology reconstructions, coding species, etc. We need fossils to date the phylogeny, to give a temporal details. And we can do biogeographical optimizations by doing species distribution coding, and then we test for diversification models. But, and this is where the barcoding story comes, we need complete species level sampling. It's not possible nowadays, with the accumulation of molecular data, with the onset of the barcoding, to actually have either generic level phylogenies or missing species in the phylogeny. And I'll try to give you a very simple illustration. So let's consider we have a group of 5 species. We have 60% sampling here, 100% sampling. We code them as being savanna or forest. Now we do reconstructions back in time. We want to understand when savanna lineages originated, for example. So in this case, whatever model, reconstruction model we use, we're going to get eventually a savanna origin for a certain time, and then here's some kind of uncertainty. Now we're missing species. If we get 100% species sampling and do the same exercise, then that's a lot more complicated. And this is quite essential. And I think that as evolutionary biologists, we should use this barcoding, DNA barcoding revolution to actually not only think about building phylogenies, but think about sampling. And this is quite crucial. So this conference, we're going to hear quite a lot about the use of DNA barcoding, and we're going to have great talks. And I'm going to try to fit evolutionary studies, the use of phylogeny within this barcoding framework. So you're all familiar with this. This is gene bank accessions, the number of accessions through time. This is the PCR. The PCR is about this time here. Now, if we look at, I randomly chose this paper. I'm sorry if I didn't cite some seminal work from people from the audience. So it's just random papers. I just took the first ones, right? And we can see basically that by early 2000, this is when this DNA barcoding kind of originated, right? And it's not direct evidence, but I'm quite confident to say that basically DNA barcoding somehow, maybe indirectly, led to the accumulation of molecular data and this rapid increase of accessions that we have available today. And I would, as an evolutionary biologist again, I would consider DNA barcoding as an effort towards comprehensive species sampling. And this is trying to build a link between, I've had discussions about this, linking applied research with fundamental research. So trying to have DNA barcoding not only helping for all these kind of things I talked about earlier, forensics, et cetera, but actually to help fundamental researchers to be able to give insights about the evolution of living organisms. Evolutionary radiations, evolution in general, this is an old concept. Simpson's 1944. We're still using these same concepts. I mean, even with the molecular revolution, we're not building new concepts. We're basically still using this. He was a brilliant guy. He talked about entering an evolutionary, an ecological opportunity leading to diversification. That's a phylogeny. Basically what it means is that you have an ancestor, that's the ecological opportunity, and you have a diversification within this ecological opportunity. Many papers have been published about this. Now there's one thing we don't know, is the actual temporal progressions of radiations. How these radiations, when they occur, how do they evolve through time? Everything is dynamic, everything evolves. So it's not the diversification per se that we need to look at, but it's how it evolves through time. And that becomes problematic. In this case, let's consider this ecological opportunity, altitude gradient, soil moisture gradient. These guys cannot move, they cannot change niches. If they occur in this particular ecological niche, they cannot move. If they get out, they die. Then the kind of dynamic you would get is a diversity-dependent pattern, simply because you have an increase in diversification, and then the niches are filled, that's it. And that's a lineage through time plot, number of lineage through time. One of the best, most beautiful example, again, is the Caribbean anoles, where there's a dispersal into Caribbean islands. For anoles, they spread to islands, they fill the niches, and that's it. And the topology of the phylogeny look very much like that. Now the other one, and this is the very difficult one, is that let's consider that actually species can switch niches. They can adapt to different ecological conditions. Now we have this phylogeny here, and you see that actually sister species, for example, the green and the pink, that's a sister species, and actually by allopatry, for example, there have been like a geographical barrier or whatever, subsequent to dispersal, then you have diversification. And the kind of pattern you would see is basically an unbounded diversification, an increase in the number of lineages through time, with nothing that seems to make it decrease or make it slow down, or is just a simple increase through time. Now this is all conceptual, so I'm going to try to give you biological examples for the two, for the bounded diversification and for the unbounded diversification, using plants, African plants, obviously. Let's look at this place here, the Cape. We're looking at 90,000 kilometers square, species rich, plant biodiversity hotspot. Most of you, I'm sure, know the Cape region. This is somehow this region we're looking at. Now, it's quite nice to work on the Cape region because there is very good paleontological record, and there is quite a lot of information we can use, especially regarding times. So the Cape Fall Mountains, all these guys here, they're old, they're Paleogene, so really 50 million years. And then we have a Miocene-Pliocene, so quite recent, exposure of coastal plains. So we have this kind of very nice historical knowledge about the actual Earth history, the abiotic evolution. And we know that there have been the onset of the winter rainfall. We're in a summer rainfall region here of South Africa, and there it's a winter rainfall, right? And that's late Miocene as well. So we have this nice time vision, right? Now we look at ratios, very cool, very nice, because they're a monophyletic group, they all occur in the Cape, and they are very species rich. So quite nice. And I'm working with Peter Linder, who is the specialist of ratios, so obviously, you know. Anyway, they are dominant in the fynbos. We find them everywhere, from coastal plains to very high mountain peaks, every altitude, every kind of ecological preferences or attributes. And we know that they have been evolving in the fynbos throughout the Cenozoic. So they're quite old. And this is the hypothesis we tested. So are there any shifts in the diversification rates of ratios? Is it linked to the Earth history we know about? Are there faster rates of diversification in different habitats, et cetera, et cetera? And there you go, 98% of total species number. So we can be quite confident on the rates we infer and on the reconstructions we do. We're still missing 2%. We are working on it. We select the best diversification model, and then we score habitats. So just to show you nice pictures of the Cape, we call it mountain wetlands, mountain drylands, coastal wetlands, coastal drylands, for all of these African ratios. We build a phylogenetic tree using fossils, et cetera, and this is what we find, unbounded. Nothing seems to restrain species accumulation through time for restoration. Now, how come? What's driving this diversification, and why there is no evolutionary constraint? It seems, I mean, there is obviously a limit in ecological space, but. And when we actually map habitats on this, we find that, let's forget about the methodological details, the best model is a kappa model, and that's close to 0, statistically significant. What it means, and I'll show you on the next slide, is that every single transition from one habitat to the other are linked to the speciation events. So each time there is a dichotomy, a speciation event, there is a transition into a novel habitat. So it's not direct evidence. This is the Gould storytelling, but this is as far as we can go. This is mountain, drylands or wetlands ancestral habitats, which fits the Earth history knowledge we have. And every single transition is linked to speciation events. And again, we were able as well to corroborate, have congruent results between the phylogenetic reconstructions and the paleontological data we have. Because if we date the origin of coastal lineages, we indeed somehow end up in this time frame of the coastal exposure. So it's quite nice. It's indirect evidences that are not correlated that kind of converge into the same story. So we have absence of density dependent, so unbounded diversification. Nothing seems to restrain these guys to diversify. And is it unusual for the Cape flora? I mean, that's a fair question. I know all these guys that I'm citing here, so you know there's no problem if I criticize a bit what they've done. It's just that they did report diversity dependence. So basically, a limit to diversification and a slowdown toward recent time. Now, all these guys did not sample 100% of the species. They all had between 15 and 55% of the total species numbers. It doesn't mean that it doesn't exist. It just means that we need more data to significantly test for these signals. The presence of unbounded patterns in the Cape. Luis Valente and Pablo Vargas published a paper in which, indeed, they looked at 19 clades, but same thing, not species-level sampling, not comprehensive sampling. And we have these habitat transitions followed by radiations, and that we can discuss the idea that it could be allopatry. So they disperse into a new ecological niche, and suddenly there's a barrier, and that's ecological differentiation, creation, I hate this word, creation of new species in different habitats. And obviously we have further questions. What about the stability of the Cape floristic region through time? How common this process is in other biodiversity hotspots, thinking about Australia or the Californian chaparral, etc. Now, the second case is a bounded diversification. So there is some kind of limit. Grasses, danthonioids. They are dominant in temperate systems in the southern hemisphere. We're looking at about 300 species. And there is a very strong spatial variation among continents. This is Africa with 122, our Australian friends with 48, and South America 48. So this kind of disparity in species number is quite interesting. Now we did the same, 90% species sampling. We're working on the 10% missing. And we did diversification analysis, and we see a slowdown. Huh? And if you look at the shifts, that's Pantamiris, that's a Cape clade, endemic to the Cape region. Well, endemic to the Cape region. No, some species occur outside of the Cape, but this is African, and this is Australian, New Zealand, South America. And we have a shift in diversification for these two guys. Now, if we look at the speciation rate through time, we see that for, follow the color scheme. So the Retidosperma, the Australian, there's a decline in speciation rate towards recent time. And same for the Africans. And all these occur in the Miocene. So is it the winter rainfall? Is it not the winter rainfall? We don't really know. So the cool thing then is to actually go and do a bit what Crystal was talking about just before, trying to identify traits or, you know, like suspect that some traits may be adaptations. Maybe, and again, this is very empirical and we don't know. That's the beauty of evolution. And basically, we looked at plant height and the degree of sclerophyll, quite a lot of anatomical work. So if you're orthophil, you look like this. If you're sclerophyll, you look like a pine. And you can do that by just doing anatomical sections. So this is an orthophil grass. You see a lot of blue, right? And this is a sclerophyll one with a lot of sclerenchyma. This is the white part of this cross-section. And we did that for this 90% species, so 279 species, and we actually were able to come up with a trait, with a quantitative trait for the degree of sclerophyll. Now we try to do reconstructions based on statistical models. It's very boring to explain, but basically we go back in the past, right? And we try to define evolutionary regimes, so modes of evolution, how this plant height evolved in the grasses, and were there different regimes, rates of evolution, faster or slower rates, and as well the kind of optimal values for these particular traits. And with the Retidosperma, here's the Australian radiation, right? There was a shift. And we find indeed that on the whole of the danthonioid grasses, only this clade here seems to have underwent a particular regime for height. If you look at the distribution here at the tips, all these bars indicate the actual value. And you see that indeed, for example, these guys here, Cotaderias, they're huge. These guys, one species can reach 12 meters, right? And these guys here tend to indeed evolve smaller heights. And yes, and the phenotype, the optimum is significantly different. So the red here represents this clade, and we see that in terms of phenotype, it's significantly smaller height than the rest of the tree. Now, if we go even further down and try to link diversification rate with the evolution of plant height, we see that within the trait here, that's, let's think about it as morphospace, the values that fall within this red area, the lineages exhibit higher speciation rates. So it's complex. There is no one single answer and one, but we have this tendency of having faster rates for smaller plants. Now, we do the same for the degree of sclerophyll, and that's very nice because then even the African guys have a particular regime of evolution. And same story than plant height. Here are distribution of the data, and just have a look at the actual variation that there is between these two guys. That's the two radiating ones. Here, Kionocloa is pretty much not variable, but here we have quite a lot of polymorphism. And again, these values here, which is a trend towards orthophily, so tender leaves, seems to be associated with higher rates. So this is the kind of work we can do. And again, there is no straight answer. There is no direct evidence. Making causalities is very difficult. So we need to make interpretations, suggestions, and make sure that we are statistically robust. And obviously, the story about the grasses, it's not the end. We know that it's a lot more complex. And if you look at this, I had fun, basically, just mapping all the events that happen for the danthonioid grasses. We have polyploidization several times on the tree. We have differential diversification rates between clades. We have dispersals between continents. We have shifts in plant heights, in sclerophyll, etc., etc., etc. All this complex picture may actually explain the diversification patterns we see today. And I think I'm done. Yeah, take-home messages. Sorry, 2 seconds. Take-home messages. As I was saying earlier, the DNA barcoding, I think it helped and is helping achieving these comprehensive species-level phylogenies. And as phylogeneticists, we should aim for this to get 100% species sampling. The development, obviously, of analytical tools. These bioinformaticians guys are doing a great job in developing new models, incorporating complexity to the models. So we both becoming statistically powerful. And when I'm talking about the new era, I think that with the accumulation of molecular data, with the power, the statistical power we're gaining, we, little by little, it's a long process, we may actually disentangle one of the complexity of evolution as a whole. And yeah, and even though DNA barcoding is used in forensics, in international trade regulations, et cetera, et cetera, et cetera, it's for us, for evolutionary biologists, it's a great, great, great opportunity. And thank you very much.
Thank you, Yanis. It is lunchtime, but I think we can engage with 1 or 2 questions.
Well, thank you very much for your talk. Like, I understand when you say we can use barcoding as a framework to gain more samples and denser sample at the species level, but I'd like you to elaborate on the concept of using barcode markers, especially for plants that we know barcoding has some limitations currently, for deeper phylogenies. And so do you suggest that we go with designated markers like RBCL, MATK, and perhaps ITS, and then just don't worry about all the other things that are happening in the Tree of Life projects and phylogenomics and so on, or it doesn't maybe matter at the species level that and the local level that you're looking at? So what is your opinion?
Thanks. This is a very good point. I would definitely step aside the idea of a universal plant barcode for plants. And I think that it's really, it depends on the context, on the groups, and no ITS, please, because dating with ITS is just, it's impossible. So sticking to chloroplast markers, getting as much variation as you can, playing around. So basically, it's really, really contextual. It depends. Grasses, for example, if you take RBCL, there is a huge rate heterogeneity in the rate of evolution, even between closely related clades. So to me, it's really, really contextual, and yeah, and we should use the barcoding data that there is without necessarily trying to pinpoint one universal marker. I think we should really be more open to, and again, it depends what we want to do, right? For this type of studies, I would really stick to chloroplast and see, and then start playing around, you know? I add this, what do I get? Does it make sense? And basically, and talking with people developing barcodes as well.
Yeah, I think we all want lunch, so I'll keep it short. You said 100% taxa sampling is very important, and I completely agree. But what about all the extinct taxa?
So, completely right. But then that's another talk. Because I completely agree. If you look at the last development, Beast 2, for example, Tanya Stadler and all these guys, they're implementing models in which we can incorporate fossils to data the phylogenies. Some people are busy trying to sequence extant taxa. So this is a whole other problems. You're making my life hard here. But I completely agree. I completely agree. If we can include fossils in a phylogeny, we have to. Unfortunately for grasses, the fossil record is ridiculous. But yeah, we should. We have to, completely.